Hi guys, welcome to Chapter 6 on Metabolism, Energy, and Enzymes. So a quick comment before we really get started. This is a super short lecture, so I just wanted to, throw it together and put it up for you, as it is not terribly complicated. But the next two on photosynthesis and cellular respiration can be pretty difficult. So when we get to those, you need to keep that in mind. There's going to be a lot of stuff, and it's really going to take you going back and forth through it a few times. So don't think that this is a real simple one and all the rest of them are as well. The next two chapters, they will require probably the most work that you apply in the class. So just keep that in mind as we go forward, and let's talk about metabolism. All right. Let's see, cells and the flow of energy. Now, again, as I said previously, the first test kind of gave us everything that we needed to do in the class, and then the second and third test just sort of build off of that, and that's what we're going to be doing here. Most of this is going to be very, very familiar to you. Okay, in the world around us there is energy being the capacity to do work, and there are two forms of energy that are worthy of our time. These being kinetic energy, or energy in motion, or doing things. And then, of course, there is potential energy, which is a storage form. And for our purposes, a classic storage form of energy would be chemical energy. We have energy stored in the foods that we eat. We store energy internally in for instance, like the glycogen in the liver or fats that we store. All of this is chemical energy. All of this falls into the realm of potential energy. So, we can store energy in large, complicated organic molecules. That's a real take-home message here. Now, before we go any further, it's important that we get out of the way the two laws of thermodynamics, at least the first two. These are the first law, and this is so important, man. You got to know these. First law of thermodynamics states let me rephrase. This is the law of conservation of energy. And it states that energy is neither created nor destroyed. It simply changes form from one form to another. Okay. This screen is on right now because there's energy coming in from the wall. That energy coming in from the wall probably came from a coal-fired power plant. My kiddo. Probably came from a coal-fired power plant here in Tuscaloosa, and that's a fancy way of saying that it came from coal, and saying that it came from coal is a fancy way of saying that it came from the sun at some point. That energy came in as sunlight, was stored into a form of plant, was compressed over time, eventually dug up by us, burned. The energy from that plant was then liberated as heat, which boiled water, which spun a turbine, and that turbine generated electrical energy that came in here and powered this screen. That is to say that energy is neither created nor destroyed. The energy from this screen, like I can feel of it, it's warm. Okay, it's radiating heat. It's radiating light. This energy, none of it is destroyed. It simply changes forms from one form to the next. And then, of course, it's the second law of thermodynamics, which plays in the role here. This is the law of entropy. And entropy, quite simplistically, means that as energy changes form, there is a net loss of usable energy. What's my exact definition? When energy is changed from one form to another, there's a loss of energy that is available to do work, or usable energy. When I crank up my car and drive from here to the college, I'm using energy stored as gasoline in my car, chemical energy, to take me from here, the house, to the college. But there is a loss of energy along the way. Not all the energy that goes into turning the wheels is going to, let me rephrase, not all the energy in the form of the gas turns the wheels. Most of it gets lost as heat. You can put your hand on the hood of the car and feel the heat coming off of it. That is entropy, folks. That is entropy. Every time energy changes forms, there's a net loss. I could flip a light bulb on. You could put your hand on it. It's radiating light, but it's also releasing massive quantities of heat. That is, as energy changes from one form to another, there tends to be a net loss of usable energy, oftentimes as heat. And that's sort of what we're getting at here. So as sunlight comes in, some of it gets sequestered by plants. A lot of that's released as heat from the plant's metabolism. You might eat that plant or eat something that ate that plant at some stage, and you can go and get a thermometer and check your temperature right now. chances are you're, around 98.6 degrees, and that is to say that you are constantly radiating heat into your environment. That's entropy. You eat a cheeseburger, not all the energy from that cheeseburger is going to become a part of you. A lot of it gets lost via your metabolically generated heat. Okay? There's a net loss of usable energy in the system. Now, let's see, what do we want to say here? Talking about energy transformations. Let me just lay this on you in a different fashion. There are a couple of concepts that you got to sort of wrap your brain around. It's hard to read this and understand it, but just try to wrap your brain around the following concept. You can have molecules, atoms, you can have atoms built into molecules in various different formats. Alright? The more complicated a molecule is, as you go from hydrogen and oxygen and carbon atoms to becoming a glucose molecule, or taking a glucose molecule to becoming a glycogen molecule, as you become more complicated, as the molecules get more and more complicated, like towards the higher end of organic molecules, they tend to store lots of energy. Okay? Lots of energy. Like the fat in your system stores copious quantities of energy. And it's really just built out of carbon, oxygen, and hydrogen. Does a carbon atom store energy for you? No, it doesn't. But if you take that carbon atom and you bind it with a bunch of other atoms and you build a big honking molecule out of it, it takes a lot of energy to make that big molecule. So in essence, what you've done by making this big molecule is you've made a big energy storage molecule. And then your body has the capacity, in the event that this is fat, to then break that down, liberating energy that you can use in your metabolic processes. This is referenced as order. Okay, you can have highly ordered molecules that store lots of energy, or you can have low order molecules, or atoms in some cases. Low order molecules that store much less energy. Energy is required to make a messy room neat. Alright, here is me in my dorm room days trying to get some studying done, and you can see that I've got junk just everywhere. If I wanted to apply some energy by getting up and organizing things, I could take all that junk and organize it into nice clean stacks. But the natural state of the world is disorder. Entropy dictates this, that the natural state of things is disorder. So if I just live in the room for a little while, even after I clean it, it'll slowly get messy and gross, as you well know. You can clean your place up and make it look fantastic, but then over time it starts to get messy again, and you have to apply energy to reorder things. Alright? The same can be said for the way that the world around us functions. Producers, organisms like plants, for instance, they take a very low order energy source, like sunlight. Okay, sunlight's quite diffuse. Producers take that very diffuse sunlight, a low-order energy source, and they pack that energy in to a very small molecule like glucose. A sugar, in the grand scheme, like glucose. They take a low-order energy source, sunlight, and they turn it into a highly ordered energetic molecule like glucose. And then you, as a consumer, are capable of eating that plant. You had a salad, eat that plant, and you absorb the glucose from that plant, and you break that glucose molecule down and liberate energy from it that you use to power your metabolism. It's all about energy storage and energy use and how these molecules are sort of assembled and put together. Yeah, that's really good enough. Let's just go here. I really want to get down here. Metabolism is the sum of cellular chemical reactions in a cell. Reactants go into a chemical reaction, and products come out of a chemical reaction. So, for instance, plants take carbon dioxide and water and sunlight. These are going to be theoretically reactants. They go into a chemical reaction, and out comes sugar from the other side, a product. Reactants are what come into the chemical reaction, and the products are what come out. Plants take carbon dioxide and oxygen and sunlight, and they kick out as a product glucose, a sugar. Okay, you will take in glucose and oxygen, and that is the reactants coming into your metabolism, and you will release as a product carbon dioxide and water. All right, this is just how this works. All right, now free energy is the amount of energy available to do a job, a task. In the realm of free energy conversation, we need to talk about exergonic reactions and endergonic reactions. This is where we are headed. Okay, exergonic versus endergonic. An exergonic reaction, is releasing something. Exergonic, releasing something. Endergonic reactions they are storing something. And what we're talking about here is energy. Okay, they are energy storing, or endergonic reactions. Exergonic reactions are energy releasing. So the book definitions of an exergonic reaction is that the products store less free energy than the reactants. They release energy. And this is a good exergonic reaction. Something simple goes in, and then, boom, crazy exergonic reaction. Heat and light billowing out from every corner. This is an exergonic chemical reaction. And what you'll find is that these release energy, and these are natural processes. These are going to occur spontaneously in cells. They release energy. If you run a chemical reaction and you feel the test tube, which we'll be seeing later, you feel the test tube and it feels like it's getting hotter and hotter and hotter. This is an exergonic reaction. If you have ever used, two-part epoxy, like resins or glues or what have you, and you mix that stuff up, you can feel it get hot before it reacts. All right, what's happening is it's releasing energy. It's an exergonic reaction. When you eat a hamburger and it comes in with all these really huge complex molecules, you can't use those. You have to break them down into more simplistic molecules. And as you break them down, they are constantly releasing energy that your metabolism uses. All right, they release energy that your metabolism utilizes. All right. And by comparison, again, endergonic reactions, these are energy storing reactions, or what we say here, they require energetic input. They are storing energy. They store more energy than otherwise. Now, let me give you an example of this. If you are making protein, muscle, okay, this is a good example. If you're making muscle in your body, muscle is going to contain huge amounts of energy. You know this if you've ever eaten a steak. A steak is a cow muscle, okay? When you are storing energy away, when you're building big organic molecules, it takes a constant influx of energy to build those organic molecules. Another example. You eat a bunch of food, and then you build fat in your system. When you build that fat, you're storing energy in those triglycerides. You're storing energy. Endergonic reactions are energy storing, exergonic reactions are energy releasing. And we can reference this by talking about the ATP cycle. So I hope that you know this at this stage, but let's just run through it anyway. ATP, adenosine triphosphate, is your energetic storage molecule of choice for immediate use. All right? You actually store very little ATP because it's so easily broken down. Recall that exergonic reactions, which release energy, tend to occur spontaneously in some cases. So you store very little ATP because it's easily broken down. All right, so you store ATP, and then you decide you need to, use your muscles, flex your biceps, stand up, walk around, do whatever you do. So you're going to use ATP to power your muscular flexion. You do this by breaking the ATP molecule down. In other words, you take adenosine triphosphate, 1, 2, 3, and you kick off the last kinetic phosphate. Boom, it's gone, and that releases energy that you can use to power your system. But that results in another molecule called adenosine diphosphate, ADP, adenosine 1,2-diphosphate. Now, you can then take ADP and rebuild this by re-energizing it, by applying energy to it, and in an endergonic reaction to build ATP. And then that will then break down by an exergonic reaction to release energy. Endergonic reaction to build ATP, exergonic reaction to release energy from ATP. Endergonic reaction to rebuild ATP, exergonic reaction to release energy from ATP. An energy flow in and energy flow out. Built by using energy from the foods you consume, release of energy used to power your musculature. That is how ATP and ADP are related to one another. This is how they function. Right, enzymes are proteins that function as biological catalysts. That's a fancy way to say that enzymes assist in chemical reactions. They make chemical reactions take place more easily within the body or within the cell or what have you. Okay, chemical reactions that would take place without enzymes happen much more easily in the presence of enzymes. And when enzymes do this, what we have is an enzyme-based substrate. The substrate is whatever the enzyme works on, and normally it's pretty easy to tell what the substrate is by looking at the name of the enzyme. So enzymes have a very specific naming convention. That is that they always end in ASE. Like for instance, acetylcholinesterase is an enzyme, ASE, that works on acetylcholine. Okay, amylase in this particular example is an enzyme, ASE, that breaks down starch molecules. These are ways in which we can identify these as enzymes, is by seeing the way in which they are named. And the real way that enzymes work is by lowering what we call the activation energy for a chemical reaction to take place. And the way I like to explain this to people is thinking about how you strike a match. So if you got a box of matches and you got a match, you can just kind of slowly rake the match across the top of the box and nothing's going to happen. But if you push hard enough and you go with enough speed, you can generate enough friction, which is heat in essence, to set off the, for instance, the sulfur or whatever in that match head. This is how enzymes work. Basically, imagine if you had a match and a box that you just barely had to touch the striking area to make the match go off. Enzymes lower the amount of energy needed for a chemical reaction to take place. If you require this much energy without an enzyme for it to happen, you require this much energy with the enzyme in place to make the same chemical reaction occur. Now, enzyme structure and function here. Let's talk about it. So, this is really simple stuff in reality. Enzymes are very specific. If you got an enzyme that breaks down starch, it's going to do nothing to glucose molecules. Okay. If you got an enzyme that breaks down proteins, it does nothing to lipids. Enzymes are incredibly specific. We call it a lock and key fit. Yeah, there we go. Lock and key fit. So they're incredibly, incredibly specific upon what substrate they are capable of reacting. They have what's referred to as an active site, and that active site will only fit whatever the substrate is for that enzyme. So enzymes are very, very specific about what they are capable of working on. An important point to be made in this is that the enzymes are not consumed by their reactions. That is to say that an enzyme can catalyze the same reaction just over and over and over and over again, hundreds, thousands of times before the enzyme gets worn out and needs to be replaced. Okay, so these enzymes are not destroyed by their reactions. They are incredibly specific, and they always end in ASE. Now, in the realm of enzyme-substrate interactions, there are what we call degradation reactions and what we call synthesis reactions. And it's exactly the way it sounds. Degradation is to break something down, whereas synthesis is to build something up. So a classic degradation reaction, we would have an enzyme here with a substrate. You form an enzyme-substrate complex, and then the enzyme does its job, releases two products in this particular case. One substrate came in, two products are released. That is breaking something down, and the enzyme survives. This would be exergonic. Okay, you're taking a highly ordered molecule and breaking it down into byproducts. That's an exergonic reaction. That is a degradation reaction. We are breaking something down, typically liberating energy that can then be used in the system. And then by comparison, there are synthesis reactions. This is where we take two substrates or more, and we use an enzyme to form these into a single product. So two substrates come in, a single product comes out. Synthesis means to make something. This is energy storing. This is making glycogen. This is making fat. This is storing energy in the grand scheme of things. Let's see, substrates join together, release a single molecule. Yeah. So energy storing, energy releasing. One molecule comes in, two molecules come out. That is breaking it down. That's degradation. Two molecules come in, one molecule comes out. That is synthesis. That's making something. And again, the enzymes are not consumed in these reactions. Now, as we proceed through this, where we're going to go is a conversation on photosynthesis and cellular respiration. And when I tell you something ridiculous like plants take sunlight and carbon dioxide and water and make glucose from it, that is so far from reality you just don't even know. Truthfully, I've been lying to you this whole time. These chemical reactions are not even remotely that simple. There are literally hundreds of minor chemical reactions that have to take place in photosynthesis to build a glucose molecule from sunlight, carbon dioxide, and water. That is a fancy way of saying that all of what we deal with in the cells, for the most part, are what we call metabolic pathways. They are metabolic pathways, and you need to be familiar with this as a concept. Cellular respiration, photosynthesis, all the reactions that take place in there to get from an initial set of reactants to a final product, there's actually multitudes, I mean hundreds, I kid you not, hundreds of minor reactions, boom, 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 that have to take place along the way to eventually lead to the final product that we think of. These are metabolic pathways. They have to proceed through several intermediate chemical reactions to get to that final product. So, in essence, you have a reactant that is catalyzed by a very specific enzyme to become some sort of product. That product then combined with other things and is catalyzed by a different, very specific enzyme to become a different product. You see where we're going here? That different product can then be catalyzed by a different enzyme with other molecules to form to become a completely different product. These are metabolic pathways. One step leads to the next step, leads to the next step, leads to the next step, and the next, and the next, and the next, until eventually you get to a final product. Cellular respiration, photosynthesis. These are metabolic pathways. You sit there and you wonder, how can this guy talk for an hour about taking carbon dioxide and water with the sun's energy and making glucose? I can do it for an hour because it's a really crazy complex metabolic pathway. Alright? It is a metabolic pathway. Alright, good. Let's see. Factors affecting enzymatic rate. Oh yeah, absolutely. So there are several, but three that we're going to talk about here are substrate concentration, temperature, and pH that affect enzymatic rate. So enzymatic rate is how fast or how slow an enzyme works. Alright, substrate concentration. Think about it like this. The more substrate there is available, the less time it takes for an enzyme to find a substrate upon which to act. If I've got a fluid with 5 or 6 molecules in there that an enzyme can catalyze, I put the enzyme in, the enzyme finds a molecule, breaks it down, and then it takes it time to find the next. And then it breaks that one down, then it takes it even longer to find the next. But what if our container is just packed full of substrate? Well, then the moment the enzyme catalyzes one reaction and breaks down, or releases, I should say, it'll immediately find the next molecule and break that one down, then immediately, and then the next, and next, and next. The more concentrated our substrate is, the faster the enzymatic rate becomes. Okay, the faster it becomes, to a point. Then there is temperature. When you increase temperature just a little bit, what this does is it increases intercellular motility. If you recall Brownian motion from the previous lecture, when you heat up a fluid, the molecules in there jostle around with greater speed. If you remember back to when I showed you the dye experiment from lab, you could see the hot water and the cold water. The hot water, all the molecules in there were just jostling around like crazy. So when we put the dye in, the dye just immediately diffused all through it. That is to say that as temperatures rise, molecules move around faster. When the enzyme does its job and breaks loose, it can find a new substrate with greater speed. So if you raise the temperature just a little bit, it's going to increase the metabolic rate. It's going to increase the enzymatic rate. But you can cause problems with this. If you get an enzyme too hot, it can begin breaking down its hydrogen bonds, and that's what we call denaturing that enzyme. A denatured protein, a denatured enzyme, this would be breaking down its unique three-dimensional structure and causing it to no longer be functional. All right, so you can heat up an enzyme to a point and have it work even faster, but then eventually it'll start breaking down and degrading from too much heat. And then last but not least is pH. So pH can also increase or decrease enzymatic rate, but this is really quite variable. Quite variable. For instance, pepsin, its pH optimum, an optimum pH is going to be where it works best, is around 2. So you can guess where in your body you'd have a pH of around 2 where pepsin does its job at most high efficiency. That would be in the stomach. And then by comparison, trypsin does its job at a pH of around 8. So that would be downstream in the small intestine, this would be in the stomach. These demonstrate that some of these enzymes work better at some pH values and others work better at other pH values. So pH can certainly influence cellular metabolic rate. Then there are, of course, other ways of activating and deactivating enzymes. A good way to activate an enzyme and make it work more efficiently is by the addition of a cofactor or a coenzyme. These can be all sorts of things, typically non-protein organic molecules, sometimes ions, very simple things, other times pretty big, complicated molecules. But all the same, when you add these into an enzyme while the enzyme is working, these will make the enzyme either work more efficiently or work, period. So without these, sometimes the enzyme can't function, and sometimes the enzyme can work without them, but it needs them to work most efficiently. Vitamins are key contributors to the formation of coenzymes and cofactors. Vitamins are the forebearer molecules. Vitamins are big old honking organic molecules that can then be used to make something like a coenzyme, which is why we say eat your vitamins and minerals. All right, and then by enzyme inhibition is where we're headed next. Cells are capable of regulating the presence and absence of an enzyme to cause the enzyme to do a job, so you can either have it or not have it. Cells are capable of regulating the concentration of the enzyme, so the more concentrated it is, the faster it will work. And cells are capable of activating and deactivating some enzymes. So enzyme inhibition would be one of the ways you deactivate an enzyme, and by adding in a cofactor or a coenzyme, this would activate that enzyme. So let's talk about inhibition. You know that enzymes have an active site. We talked about this previously. So enzymes have an active site. So you've got my enzyme here, and the active site is the area where it will attach to a substrate and then either break that down or build it up into a product. So enzymes have an active site. In the realm of inhibitor molecules, there are what are called competitive inhibitors and what are called non-competitive inhibitors. A competitive inhibitor is one that can bind to the enzyme's active site and block that active site. So the inhibitor will bind in to the active site, and while it's there, no substrate molecules can come in and bind. Ergo, that enzyme is non-functional. That is competitive inhibition. The inhibitor competes for the active site with the substrate. And by comparison to this, there's also non-competitive inhibition. Non-competitive inhibition is when you have an inhibitor and it binds to some other location on the enzyme that stops the enzyme's active site from working anymore. A lot of the time we call this an allosteric site. Allosteric means other area or other part. Okay, so here we have an enzyme and it's got an active site here. No, wrong. Here we have an enzyme and it's got its active site here. It binds to this molecule and breaks it down. But if you bind an inhibitor to its allosteric site, it no longer is capable of binding to its substrate. So a non-competitive inhibitor binds to a non-active site part of the enzyme, which deactivates it. And a competitive inhibitor binds to the active site to prevent that enzyme from then doing a job. All right, sometimes competitive inhibitors can be, or let me rephrase, let's just keep this simple. Sometimes inhibitors can be a real problem. Sometimes they can bind irreversibly and destroy that enzyme in essence. They make it non-functional forever. It will never ever work again, and these are called poisons. Things that you've heard of, like cyanide, sarin gas, warfarin, sort of famously, are going to be enzyme inhibitors that can literally cause you to die very quickly. Cyanide is a really famous one of these, okay? Cyanide comes from bitter almonds, and if you've ever watched like a movie where somebody got poisoned, they start frothing at the mouth and just fall over dead, that is classic cyanide poisoning. What cyanide does is an inhibitor, it inhibits the enzyme that makes ATP. Okay, cyanide inhibits the enzyme that makes ATP. So imagine you have been exposed to cyanide and you take a breath, then you release it. Well, you just used all the ATP that power your diaphragm. So the next time you take a breath, you don't have ATP that allow your diaphragm to relax, so now you're going to asphyxiate. You're going to die from the inability to breathe. And as you're writhing around trying to, figure out what the heck's going on, your muscles all lock up in unison because you don't have any ATP to release those muscles from their flexed positions. That is cyanide poisoning, and a rough way to go. That's a real bad way to go. Believe it or not, snake venom does a very similar process to this. Snake venom, let me just jump over to here. Snake venom gets in and blocks the receptors from your neuromuscular junctions. So, in essence, think of these as enzymes to some degree. It gets in and blocks them from being able to be stimulated by your nervous system. So your nervous system is sending messages down to your muscles, but your muscles are incapable of responding to that neurotransmitter. That causes the muscles to lock up, and you become paralyzed, and you stop breathing, and that's how you could die from a snake bite. Or alternatively, if a snake bites like a little rabbit or something like that, they will become paralyzed very quickly, and then the snake's capable of consuming it. These would be sort of examples of poisons, of, what do I want to say here? Inhibitors that permanently bind to enzymes, rendering them no longer operable. No longer operable. Yeah, that'll work. So incredibly deadly. Incredibly deadly. And I think that's it. Yeah, that's it. So let's stop there and expect a lecture on photosynthesis a little later this week. And it's going to be interesting. These are big, long lectures. The next two are complicated, so be ready for it. All right, thanks, guys.